In this module of the MIQE series, I'll present data that demonstrate the effect of sample-derived inhibitors on the quality of final qPCR data. The MIQE guidelines were published in Clinical Chemistry in 2009. The MIQE paper describes each of the steps that are required for validating the processes within a workflow that takes you from analysing the quality of the samples to analysing the quality of the assay. And really only when those two steps have been completed should the assay and the samples be used together for target quantification. Within this seminar, we'll examine in more detail the influence of inhibitors present within samples on the final qPCR data. There have been various methods described for the identification of inhibitors within a sample. One of these we refer to as the SPUD assay, and this has been published by Nolan et al. In Nature Protocols. The assay works by construction of an artificial template labeled the SPUD template. To this are directed two specific primers and optionally also a hydrolysis probe clearly within the location of those primers. So when this assay is run in the presence of PCR buffer and in the absence of any target, there will clearly be a diagnostic CQ dependent upon the concentration of the target that's been included. In the presence of sample that does not contain any inhibitors, you would expect to record exactly the same CQ as you do from the reaction in the presence of only buffer. However, if the reaction is run in the presence of an inhibitor carried through from sample, and in the example shown on the left here, we can see the presence of phenol left over in a sample from the extraction process. Now the assay has been inhibited. We're still detecting the SPUD, but we're detecting the SPUD at lower efficiency because it's inhibited by the sample. On the right-hand side, you can see a sample that apparently, according to the adjuvant bioanalyzer analysis, is of high quality. However, we know that during the preparation of this sample and one similar, a concentration of EDTA was added, and this was done to simulate the effect of an inhibitor within a sample. And you can see that the effect of the EDTA on the SPUD assay is to inhibit at fairly low concentration by 2 cycles. When you double that concentration, you can see that the assay is completely inhibited such that it fails completely. So this also indicates that the effect of an inhibitor in a sample is not linear. So where on the previous slide we observed that the effect of an inhibitor in a sample is not linear in terms of CQ recorded from a qPCR, in this example we're examining the effect of the same inhibitor at different concentrations on different assays. And here you can see that the mitochondrial assay, shown in red, is much more sensitive to the presence of presumably urea in the urine sample than is the SPUD assay. So now we can see that the presence of the same inhibitor in an assay has a different effect on different qPCR assays. So therefore, when you were performing a normal experiment of, for example, measuring one concentration relative to another, as in a normalization, you can see that the presence of an inhibitor would completely destroy the real ratio you were expecting to record. And of course, it would be reasonable to expect that this effect wouldn't be too much of a problem if using a well-defined purification procedure. Well, in this example of a set of data, again provided by Jim Huggett from UCL, you can see quite clearly that using any of the well-recognized purification processes, none of them were able to absolutely remove all traces of inhibitor from, again, DNA extracted from urine. So it's pertinent to remember that however well you believe your extraction process has been at removal of inhibitors, it's important to check the sample and ensure that they are pure. And while those examples are demonstrating the effect of an inhibitor that is within the sample from its environment or from its source, the paper shown here demonstrates that even the reverse transcription remaining within an RT, which is generally remaining bound to the cDNA, will then have the effect of inhibiting PCR, particularly when at low concentrations of target. So, of course, this is particularly important if you're working with, for example, single cells or very small numbers of cells or laser capture samples, for example. And it's important to recognize that one step that can be taken to remove this is to perform an additional ethanol precipitation in the process of ammonium acetate. But again, it's important to verify that the sample does not still contain inhibitory factors. Jim Huggett's work went one step further to then examine the relative effect of RT enzyme on different templates or different sequences of template. So here you can see the effect of 50 units of RT within a reaction on the SPUD template, whereas in this data you can now see the effect of the RT enzyme on a 16S assay. And you can see that the 16S assay is much more sensitive to the remaining presence of RT within an assay. Once again demonstrating that different assays have a different sensitivity to the same inhibitor within a sample, and of course meaning then that relative quantifications of different targets will be perturbed in the presence of any inhibitors. So then we've observed that inhibitors can arise from the sample purification process within the storage buffer, we showed EDTA, and even the RT enzyme itself. Different inhibitors have a different effect on different sequences. So this means that if there is an inhibitor within the sample, it is not viable to simply normalize away the problem by assuming that all assays are affected to the same degree. So for accurate quantification and to ensure that you do not have false negative results, it's important to detect and either remove or dilute the sample to dilute out the effect of the inhibitor. The data that I've just demonstrated were published in BMC Research Notes in 2006 by Jim Huggett. Most of the data that we've just demonstrated was concerning the effect on either DNA or RNA-based qPCR assays and looking at the effect on the enzymatic process of amplification. However, in these examples, we have a slightly different effect on the assay. Here you can see an effective genotyping assay being performed on DNA extracted from whole blood. Very clear differentiation of the genotypes. However, these are the data that are derived using exactly the same assays, only in this case it was unfortunate that the sample had been allowed to lyse prior to the genotyping experiment. So clearly we can expect that there would be the presence of heme remaining in the purified DNA. So on the previous slide, we saw that the presence of heme was seriously impacting on the quality of the genotyping assay. And after a series of studies, we recognized that it was the presence of heme that was affecting primarily the hex assay. So in a comparison of the effect of the heme with either FAM dye or the sample, the heme, in the presence of hex dye, you can see that by comparing the fluorescence signal from a dilution of dye in the presence or absence of the sample, the effect of the sample on the FAM signal is to enhance the fluorescence, whereas the the effect of the sample, presumably the heme, on the hex dye is to suppress the signal. So in this example, we can see that the inhibitor effect is actually an effect directly on the fluorescence signal. And of course, this means that if the FAM signal is enhanced and the hex signal is suppressed, clearly the presence of any heme within the sample, any other factor affecting fluorescence, is going to impact on the genotyping assay in the way that we saw on the previous slide. So in summary, it's absolutely critical to use the best quality template whenever possible. It's important to QC all of the templates or the samples that are going to be used, and recognize that if there are any factors within the template that mean that it isn't the best quality, then to recognize that this will cause compromises within the assay. The SPUD assay has been demonstrated. This is a universal assay, apart from for those people working on potatoes, because it's a potato-derived sequence. Otherwise, it's a universal assay for the detection of inhibitors that could potentially interfere with a qPCR assay. Throughout this seminar, I've described a range of qPCR experiments. Sigma-Aldrich has a range of products that have been developed specifically for the qPCR scientist. We have a wide range of custom oligos, including all of the qPCR probe formats, and these can be labelled and quenched with any combination of fluorescent dye or quencher that you would choose to work with. And these are compatible with all of the qPCR instruments currently available. We also have two ranges of qPCR master mixes, both of which incorporate cybergreen or are developed without for the use with any of the probe technologies. There's the regular mix, or the regular range of mixes, or the luminosity range that have been developed to be compatible with the fast cyclers. During this seminar, I've described the factors within the MIQE paper that relate to assay design. You can download your own copy of the MIQE paper from our dedicated website at sigma.com/MIQE. And if you would like to have an assay designed for you, you can request a custom design through our Oligo Architect service at sigma.com/probe design online. There's also a tool there for you to design your own primers and probes. For other inquiries, please do contact us directly at oligotechserve@sial.com. This seminar was one in the series of BioExperience Sigma Global Education Programme.